Fantastic. I'm delighted to be here and to see such a packed house this morning. 30 minutes to really update you all on the latest, greatest in Alzheimer's disease, at least as I see it, is a daunting task. Nonetheless, we're going to take a whirlwind tour of kind of where we are in the field. And there are many upsides to that, but I'm also going to show you some of the downsides to that. The problem is much, much bigger than we originally have estimated. So with that, we'll begin. I have a few disclosures here, nothing that directly is linked to this talk other than my concluding slides. I will show you some data from a compound that has been developed by Altec that we've been bringing into humans for the first time, and we've completed a phase 1 trial and have an active phase 2 ongoing. So I think that may be relevant. So in the field, we've really learned about the disease. We've learned about a progression of disease that may extend back even 2 decades before one develops memory and thinking problems, with early emergence of amyloid leading to tau, neurofibrillary tangles, and cell death. And so when we're talking today about drug discovery and Alzheimer's disease, I want you to keep an open mind. We're not just talking about the end stage of Alzheimer's, someone that we might see in a skilled nursing facility, and each one of us could diagnose from across the room. We're talking about perhaps you or me with Alzheimer's disease. And so our investigations cross the spectrum for preclinical disease all the way to Alzheimer's. And I showed just a few slides here of active research studies that are ongoing. We cannot test one agent at a time. We cannot take 15 to 20 years to come up with better treatments and cures for Alzheimer's disease. We need to be doing this in bulk if we're going to tackle this problem in time. Basic primer. Alzheimer's disease is a medical illness. It is not just the symptoms of memory loss. It requires not only nerve cell loss and brain injury and destruction, but requires these pathologic hallmarks. We've known this since Alzheimer published his first paper in 1906. There are characteristic tangles. These are abnormal accumulations of protein inside cells that are killing them. And outside the cells, to the right on the screen, are the amyloid plaques that characterize Alzheimer's disease. They're highly toxic, killing nerve cells from the outside in. And we've been able to work through this with new discoveries that I'm going to show you today to tease out the biological progression of disease. It doesn't just happen all at once unless you've had a major stroke that you would develop dementia. It is a progression. So this slide sort of summarizes that. And the first thing that we see early in the disease course is an elevation in amyloid, where it begins to deposit in the brain in the form of plaques. That is rapidly followed by abnormalities in nerve cell functioning, what we can visualize with FDG PET, followed by death of nerve cells, neurofibrillary tangles, and only over time will we see shrinkage of the brain that eventually becomes severe enough to cause memory and thinking problems, and the functional decline characteristic of what we commonly recognize as Alzheimer's disease, but again, a process. So over the last several years, the field has been very active in redefining the diagnosis of Alzheimer's. And what we classically have thought of as Alzheimer's disease is this dementia phase on the far right of the screen, where there is amyloid plaque in the brain. There are neurofibrillary tangles. The brain is beginning to shrink. There are cognitive changes and functional decline. But now we can define the disease clinically as well as a prodromal phase, where we're having amyloid abnormalities, tangles, mild memory problems, but yet no functional decline. These are people like you and I that are engaged in everyday life. And even further back to the preclinical phase. This is kind of for all intents and purposes, like discovering a polyp on colonoscopy. Is it a cancer yet? No, but is it going to become a cancer if we don't remove it? Yes. So this early preclinical phase characterized by that amyloid deposition. So I'm going to simplify that graph and that structure with this characteristic slide. And we're going to take this one step at a time, the discoveries that have been made, the new medicines that are coming out in active trials currently, and we're also going to answer perhaps the most important question of the 20th century. Did Reagan have Alzheimer's disease when he was in office? So I think that President Reagan's lifespan and his battle with Alzheimer's disease actually fundamentally paralleled some of the major discoveries in the field. So 1982, the first time that President Reagan graced the cover of Time magazine, there was no consensus. There was no clinical diagnosis of Alzheimer's disease. It wasn't until 1984 that amyloid was discovered as a core component of the plaques in the Alzheimer brain, and it wasn't until 1984 that we began to galvanize as a nation, and the Alzheimer's Disease Center program was established by the National Institutes of Health. So here we are, number one, amyloid plaques. We talk about preclinical Alzheimer's disease because we've developed ways with PET scanning, radio-labeled ligands that we can inject into people's veins intravenously. Let this circulate, it will bind to the amyloid plaques in the brain, and we can see in the brightly colored brain sections to the right an Alzheimer brain lit up in pink, and that is chock full of amyloid plaques. In a living person, something that we previously could only do with autopsy, we now can see this. And to the far left on that panel, we see somebody who is PIB-NC. That's a normal control. And that was what we expected. What we didn't expect is the middle brain. That is a normal control whose brain is already full of amyloid, compensating for that disease. So if we look at preclinical Alzheimer's disease, we look at the prevalence of Alzheimer's. This is clinical Alzheimer's disease. It begins to skyrocket after the age of 65. If we look at autopsy data, however, we can see that those plaques are building up in the brain, skyrocketing after the age of 45. And we can see that that PET scanning exactly parallels what we see at autopsy, showing us for the first time in living human beings the course of disease that is characterized by the development of amyloid plaques 15 years prior to a diagnosis of Alzheimer's. This is an important stage and an important target for intervention and treatment, and the field has been actively working towards this goal for many, many years. It still remains a primary target for disease. Despite what we often hear on the news, there have been some failures, but also discoveries as we move forward scientifically. So some of you may know that back in the late 1990s, we developed a vaccine for Alzheimer's disease and began giving it to folks. And it was very promising. In the top panel, we have a mouse model. At 18 months, the brain is full of amyloid plaques, but if we immunize those mice, they were resistant to the disease. That was the impetus to bring this forward into humans, and we did so, but unfortunately, the brains of about 6% of these subjects developed an encephalitis, an inflammation related to the vaccine that caused the trial to be halted prematurely. Since then, we've been working in the area of passive immunization. Instead of giving someone a vaccine that would just do whatever it wanted in the body, carefully controlling antibodies that we give to folks. And really, I think, this is one of the major discoveries in the second panel on passive immunization. This is amyloid PET scanning of a subject in green with red amyloid plaques in the frontal lobes at entry into a clinical trial where they received antibodies against amyloid, and 18 months later, that amyloid essentially cleaned out of the brain. The first the first evidence ever that we can change the disease process itself biologically. Now, unfortunately, even these passive antibodies that we can control can lead to brain inflammation, what we call amyloid-related imaging abnormalities. That really has been a problem in the field because very successful antibodies that are removing the amyloid are also creating problems. So we need to move forward not just with more effective treatments, but with safer treatments. Turns out that there are a host of different antibodies that can attack the amyloid cascade in many different areas. So some of our antibodies attack just the plaques. Some antibodies attack just the precursor protein, and some will address intermediate forms of amyloid. This is where the field exists today. There are about a half a dozen antibodies that are in active clinical trials that range in terms of their amyloid target, looking at different molecular species. And also, vaccines have returned, active vaccines, safer vaccines that lack T-cell epitopes to prevent the inflammation we saw in the original vaccine trials. DNA vaccines that are designed to be safer, to reduce that inflammatory response, and yet target one of the key early features in Alzheimer's disease. We're bringing these antibodies forward also with the recognition that amyloid plaques are building up 10 to 20 years before one has memory problems. We need to begin to use these in folks who are still normal in memory and thinking to prevent the later stages of disease. Another active area of research discovery is what we call beta secretase inhibitors. And the Alzheimer or the amyloid precursor protein is a transmembrane glycoprotein. It's found in all of us, in many, many tissues in the body, but most heavily expressed in the brain. In order to generate the amyloid in plaques that we see in Alzheimer's, this has to be abnormally processed. It's cleaved by two different enzymes. First a beta secretase and then a gamma secretase releasing that fragment. Now, we've tested gamma secretase inhibitors, and there were quite interesting studies. One of the major side effects of gamma secretase inhibitors was it turned some people's hair pink. Can you believe that? We still don't have a real good explanation for that, but it was hard to blind the investigators when their patients came in with pink hair. Unfortunately, there were a lot of side effects to the gamma secretase inhibitors. It's ubiquitously expressed in the body and affected many different organs. And so gamma secretase inhibition has essentially been eliminated from our therapeutic portfolio currently. But the beta secretase inhibitors are showing active promise. There are several different beta secretase inhibitors out there, from Eisai, from Lilly and AstraZeneca, Merck, Novartis, all in phase 2 trials currently, moving into phase 3, which is bringing us closer potentially to getting some of these out. These will block the production of that abnormal amyloid and hopefully prevent plaque formation. We can see in the graph on the lower right a reduction in plasma A beta, and we can see similar changes in spinal fluid as well. Treated with a BACE inhibitor for just 14 days, we could see a dramatic decline in the production of that abnormal amyloid. So this is an area of active interest, one that we're watching with great interest. So 1986, Reagan again graces the cover of Time magazine. It was in this year that tau was discovered as the major protein in neurofibrillary tangles. So we think that we've known about Alzheimer's for over 100 years, but really these discoveries are in the last several decades. 1986, the original National Institutes program was expanded in the consortium to establish a registry for Alzheimer's disease, global initiative to get doctors and scientists all using the same nomenclature, the same terms, the same diagnoses to characterize Alzheimer's disease, to put us all on the same page. So here we are moving a little bit further into the disease with tau and neurofibrillary tangles. Major discoveries over the last several years, and this comes from 2012, the discovery that once tangles start in the brain, they will pass from nerve cell to nerve cell. Almost what we consider a prion-like propagation, almost infectious from one nerve cell to the other. Now, Alzheimer's is not infectious. We cannot catch it. At least I haven't caught it yet from any of my thousands of patients. But from nerve cell to nerve cell, this tau is passed, and it was demonstrated in animal models that expressed abnormal tau in the entorhinal cortex where neurofibrillary tangles begin, that if you followed by 12 months, the abnormal tau would start showing up in the next set of neurons down the line. And by 18 months, tangles were developing, and it was spreading further into cortical regions of the brain. This has really opened up the door and explained why the disease, once started, progresses relentlessly. We've been unable to see this as well, except in animal models and at autopsy, until the last 2 years, where folks have developed anti-tau or neurofibrillary tangle PET scanning for the first time. And those are the brains that I'm showing you here. We have widespread tau pathology beginning in the medial temporal lobes, in the posterior cingulate cortex. We can now track this as well and watch its progression nerve cell to nerve cell. So what about targeting tau? This is an area of extreme interest, and the first tau agents are really just coming into trials currently. We've done this in animal models. That's the top panel. We can take an animal model of neurofibrillary tangles. We can treat it with antibodies against tau and essentially eliminate this pathology from the brain of these mice. These studies are moving forward. Other studies that have been out there are in active trials trying to prevent tau and neurofibrillary pathology. And I show some data here from an agent that's been developed by a company, Tau RX. It comes from methylene blue. Methylene blue is an agent that was given to people with chronic urinary tract infections as far back as the 1930s. It actually turns out that this prevents the abnormal aggregation of tau that forms neurofibrillary tangles. The preliminary data presented on this suggested an inverted U-shaped dose-response curve, and active phase 2 and phase 3 trials are underway currently globally looking at this agent, trying to find the right dose, seeing if it can stop this tau pathology. Excuse me. The field, though, isn't stopping there. We really need to target this. So if we miss people with early amyloid accumulation, we've got to stop the disease at this next stage. One of our active trials focused on Fin kinase inhibition, and the Fin kinases are very, very important in the pathogenic cascade of events that occur inside nerve cells. So in the cartoon figure, we see amyloid oligomers, toxic oligomers. They're activating this glutamate receptor, which then activates Fin. Fin hyperphosphorylates tau, leading to neurofibrillary tangles and Alzheimer's cell death, a loss of dendritic spines. And with a Fin kinase inhibitor, we can completely prevent this in the animal models. This is currently in phase 2 human trials. We can look further at metabolic dysfunction. We know that insulin resistance within the brain can exist without diabetes. We know that this insulin resistance leads to depletion of internal energy stores and eventual cell death. We can see this by PET scanning, the brain turning down in a patient with Alzheimer's, and we're actively investigating ways to restore this energy metabolism. Right now, phase 3 studies are ongoing across the nation using intranasal insulin to turn back on areas of the brain, specifically shutting down in Alzheimer's disease. We're looking at the receptor for advanced glycation end products as a mechanism for targeted cell death in Alzheimer's disease and inhibiting this. Actually, several years ago, probably about 8 years ago now, we ran the first trial of a RAGE inhibitor. It was stopped because of adverse side effects and a futility analysis that suggested it was not going to succeed. After we gathered all the data, it actually worked. It looked like it worked. And this is back in phase 3 trials currently targeting different molecular mechanisms for disease. When enough nerve cells die, we begin to see the brain shrinking, brain atrophy. We could see these changes 5 years before the onset of memory and thinking problems. We are actively trying to prevent this nerve cell death, and this is a compound that's been developed by Toyama Chemicals, a subsidiary of Fuji Films. Who would have thought the film industry would have gotten into Alzheimer's, but they have. This agent seems to prevent amyloid-related neurotoxicity in the figures up top and restores normal electrophysiologic function to Alzheimer nerve cells. This is currently in phase 2 trials, which should complete by November. Plans already to take it into phase 3 by early spring of next year. We're working on genetic manipulation, targeted adenovirus vectors to restore nerve growth factor to prevent nerve cells from dying and that brain from shrinking. And one of the first studies was completed, small trial of 10 here in the United States. The phase 2 trial is in preparation currently. We were able to introduce virus and restore nerve growth factor levels to normal, preventing cell death in these Alzheimer patients. So 1990, Reagan again graces the cover of Time magazine. This is when we began to recognize early memory problems preceded your life falling apart. It was when genetic discoveries began to be made, and also a hallmark year, it's when I began researching Alzheimer's. So this is what we call mild cognitive impairment. And we recognize this because if you have early memory problems, you're at high risk to develop Alzheimer's disease. The average person over the age of 65, 1 to 2% risk per year. If you have mild cognitive impairment, that is increased tenfold, 10 to 20% risk. That brings us to a point where this phase of mild cognitive impairment is going to last about 4 years before your life falls apart. And in 1994, Reagan addressed the nation, informing us that he had been suffering from Alzheimer's disease. So Alzheimer's disease as we know it, we're not forgetting about this group of patients. There are active studies underway looking at things like the 5-HT6 serotonin receptor. Antagonizing this receptor increases neuronal synaptic transmission. So new ways that we can symptomatically help Alzheimer patients function in very important ways. These trials are early. Phase 1 and phase 2 trials show tremendous promise. These are moving into phase 3 studies currently. So 2001, 7 years into Alzheimer's disease, Reagan does not even know who Nancy is at this moment by name, but he knows exactly who she is. She is the love of his life. AD, a brain destroyed. If we do not move these agents forward, if we do not stop this pathway, Alzheimer's is universally fatal. So President Reagan eventually passed away in 2004, and if you've been paying attention, you should know the answer to the ultimate question of the 20th century. Did Reagan have Alzheimer's when he was in office? So we'll track this backwards. 2004, Alzheimer's has a typical 10-year course. 1994, MCI, early memory problems, has a 4-year course. 1990, onset of Alzheimer's, coincident with him stepping down from office, his presidency ending. But we know from what I've shown you today and the discoveries that have been made that his Alzheimer's actually began in 1979, long before he even entered office. So the question, did Reagan have Alzheimer's when in office? Medically, biologically, yes. Clinically, no. That gives me some comfort that an Alzheimer patient didn't have their finger on the button. So when we look at the size of the dementia problem, this is what we see. The green circle is the population of the United States. I apologize to the international audience here. I'm not showing the world, just the United States. 13% of that population is over the age of 65, 39 million people. We blow that up into the blue circle and we see 13%, 1 out of 6 over the age of 65 have Alzheimer's, 5.2 million. But I've already told you, this is people whose lives have fallen apart. Let's look at what those figures look like if we include those with mild memory problems. Now, 29% of that population is affected, 11.5 million people. And let's look further at those that have amyloid plaques and don't even have memory problems. We can now see the size of the problem. It's much bigger than what Dr. Power showed us earlier this morning. In the United States, 62% of those over the age of 65 have amyloid plaques in their brain, irrespective of whether or not they have Alzheimer's. This is a major, major problem, 21 million people. Now, I promised that I'd show you a little bit of the data from our initial phase 1 trial. This is AT001. Some know it by the name of Cellplex. Taken into animal models of Alzheimer's disease in the top panel, this is highly effective in reducing that pathology. A yeast form of selenium that's quite unique. In humans, we can see excellent absorption, beautiful dose response curves, PK values in blood and in spinal fluid in these patients. We took this to completion, 12 weeks of treatment. Everybody donated spinal fluid, donated blood, urine. We could see a dose-dependent decrease in prostaglandins, systemic inflammation in the body. And in the brain, we didn't quite hit significance, but a small sample. It remarkably stabilized amyloid production that was changing for the worse in the non-treated elderly, even in this small study. I think it's quite remarkable. Key concepts in my closing slide, I think, to keep in mind when we look for a cure for Alzheimer's disease. We have a lot out there that I've shown you today on that whirlwind tour. We need to remember and always keep a focus on, we need to treat millions, hundreds of millions. We need an excellent safety profile. We can't go back and make the mistakes of the past, of the early Alzheimer vaccines. We need practical considerations. We need pills. We need tablets. We need some way to administer this drug. We need to target the disease very early for prevention, not wait until the brain's destroyed. We may need multiple agents, or we may need agents with more than one particular target. Agents that we sometimes consider dirty may be actually more effective than clean, pristine medications. And we need to do this now. This is really an impetus for us if we are going to save not just the nation, but the world. I'll end there. I hope I didn't go over by too much. Fantastic. Thank you so much for your attention.
Sure that there must be lots of questions. So we're going to have 5 minutes for a few questions. Yes, sir.
I'm wondering, in relation to heart disease, you talk about, Mark talked about yesterday about the sugars and stuff. I wonder if there's been any research done on if our diet, the American diet, has anything to do with Alzheimer's versus just the brain injury.
Yeah, this is a great question, and absolutely. We know that there are dietary mediators as well as many nutritional supplements that have been tested in Alzheimer's disease as well that show tremendous impact in the animal models, and that in human studies, retrospective epidemiological studies seem to show dramatic benefit. So a Mediterranean-style diet, rich in vegetables, fish, low in red meats, and low in high cholesterol foods, may delay the onset of Alzheimer's by 3 to 5 years. Some of the nutritional supplements really seem to have an impact in the disease state as well. Some of that may be genetically mediated, who is going to respond best. We haven't yet gotten to the point where we can conduct rigorous trials of nutritional intervention, but we need to go there, and that's an area of active pursuit. The problem is the pharmaceutical industry isn't going to move nutrition forward because you can't patent nutrition and good health. And so that's really left us with a hole in what we're doing research-wise, dependent on small levels of funding from the National Institute of Alternative and Complementary Medicine, looking at small companies that are interested in moving forward their nutritional products. Japan is just exploding with their brain health nutritional supplements and research, but we need to do much more. That's an excellent question. Thank you.
Excuse me, sir. I'm from Brazil, and I come from a family that everybody dies from Alzheimer. And I also have a nephew that he has the Down syndrome. Is there any relation between these two diseases?
That's a great question. And actually, we have an active research program funded by the National Institutes of Health looking at adult Down syndrome. You know, Alzheimer's does run in families, and there are true genetic forms of Alzheimer's. It represents only about 1% of worldwide Alzheimer's disease. Colombia, South America, one of your neighbors, actually has the largest family of autosomal dominant Alzheimer's disease. So there are clusters where this runs. Typically, if it's autosomal dominant, you're going to come down with the disease in your 40s or 50s. Very, very early for age of onset. It's a very characteristic. If any of you have family members with an early age of onset like that, you should be concerned. Those mutations all work through increasing amyloid. There are 3 known autosomal dominant mutations. Now, fascinating, there's actually a family in Iceland that has a reverse mutation. They are immune to Alzheimer's disease. Isn't that wonderful? Boy, I wish I had some Icelandic blood. When it comes to Down syndrome, we know that Down syndrome is caused by triplication of chromosome 21. The amyloid precursor protein exists on chromosome 21, and that increases amyloid production throughout a lifespan in patients with Down syndrome. Universally, by the age of 40, every person with Down syndrome has a brain full of plaques and tangles. And as a matter of fact, the autopsies on children that have died from some of the cardiac manifestations of Down syndrome have shown amyloid plaques in patients as young as 6 months of age, babies who already have Alzheimer's disease. Absolutely amazing. So we need to work for this, but really it's provided tremendous insight into this pivotal role of amyloid very early on, initiating the degenerative cascade.
Time for one more.
Question. You talked about the insulin-resistant brain. How would this relate to women with PCOS who are insulin-resistant? Would they have an increased chance of brain disease?
That is a great question, one that I don't have the answer to. We do know that diabetes, especially type 2, insulin resistance systemically, however, is a risk factor for Alzheimer's disease. So one would assume in PCOS, where insulin resistance is quite high, that they would carry that same risk for increased incidence and prevalence of Alzheimer's.
Even without the actual disease, diabetes?
So even folks without the actual disease, insulin resistance shows a tight association between Alzheimer's and degree of insulin resistance. the more resistant you are, the higher your risk for Alzheimer's.
Okay. All right. Well, thank you very much, and thanks again, Dr.
Fantastic.
Jicha.
Thank you.